Hi everyone, I'm going to start the task assigned. So let's see the instructions. First, we need to do a pre test in 20 minutes. So let's set the timer for 20 minutes and let's do the test. This is the test. A biology, a cell diagram has been given, and by using this diagram, we need to answer the following 6 questions. Let's see. Label G represents several proteins that assist with cell division, protection, and maintenance of cell shape. What do these proteins comprise? The G is label F represents dash, and then the organelle represented by label D is mainly found in label D is mainly found in, as I don't have any idea about this, so let's put Na. And the main function of the Golgi apparatus, I think L and K has been given. I think it's K. Sending and receiving proteins. The organelle labeled C is present in, let's put the animal cells only. The organelle labeled dash will be in higher numbers in muscle cells compared to the fat cells. Let's say H. And then next, just as an amino acid is a monomer of a protein, a dash is a monomer of DNA. Oh, let's see this question. A DNA monomer is called a nucleotide, which means we get this answer, no? A nitrogenous base, deoxyribose sugar, a sulfate group. Which part is correct? Incorrect. DNA is more stable. Then let's see the next questions. There has been given another diagram. Use this diagram to answer the following 7 questions. According to the strandedness of this molecule, what type of nucleic acid is it? I don't have any idea what is this. So that's for DNA. What type of base? What is paradoxical about this image? I don't understand what is this, so let's put DNA or something. According to the question 11, sigma and epsilon. I don't know, so let's put this like that. AU. And for this, let's reverse it. Again, another diagram has been given here. Use this following diagram along with the previous to answer the following two questions. It looks like some carbon compound. Let's see the question. This is the sugar from the previously discussed nucleic acid. There is a hydroxyl bonded to the 2, 3 and 5 carbon. What type of nucleic acid is it? 2, 3, 5. I don't have any idea about this. What conclusion can be drawn from your findings? Let's put A. Let's see the next questions. The ordinary flow of information in a cell is DNA, RNA, and the last one I don't have any idea. The process that takes place at the first arrow and the process at second arrow is, I think, transcription and translation. Which statement about proteins is correct? An alpha helix is indicative of primary structure. Let's select an alpha helix as indicative of primary structure. Let's see next questions. Use this diagram to answer the following three questions. A DNA template strand contains the sequence. What is the complementary? Let's select A. Your mRNA strand of choice must be reserved before converting to amino acids. That means it must be. Let's select this from 5 to 3. What protein sequence does this mRNA code for? Notate each protein as its 3-letter abbreviation. See, GAG. GAG is glue. Glue. And then AAC. AAC is here. ASN. And then TAT. What is this TAT? Until then I figure it out. So let's skip this. A point mutation causes an amino acid to change to a stop codon. What is the effect of the resulting polypeptide? Let's put the truncated and non-functional. A second point mutation causes that to stop codon to change into a codon that is different from the original. What is this mutation called? I think it's missense. I had finished the. Let's submit this and let's check the score. 9 out of 5. The first one is correct. Label G, cytoskeleton, and label F represents the centrioles and cell division. The rough ER. Okay. The main function of Golgi apparatus is sending and receiving. Organelle labeled C is present in both animal and plant cells. The organelle labeled will be in higher numbers in H. I think it's the mitochondria. And yeah, the next it's amino acid. A nucleotide is a monomer of DNA. And for DNA, nitrogenous base, deoxyribose sugar, sulfate group is the wrong one. DNA is more stable but less reactive. Okay. And then, according to the standardness, nucleic acid is DNA. And according to these types of bases in the molecule, what type of nucleic acid is it? It is RNA. What is paradoxical about this image? Double strandedness is indicative of DNA. It's AT. Okay. What bases do? Sigma and epsilon represent AU. Using this following diagram along with the previous to answer the following two questions. For this, the first question, this is a sugar from previously discussed nucleic acid. There is a what type of nucleic acid is it? I put DNA, but the answer is RNA. What conclusion can be drawn from your findings? RNA can sometimes be found as double stranded. Okay. The ordinary flow of information in a cell is DNA, RNA. The final one is protein. Okay. The process that takes place at the first row is, and the process at the second row is the correct answer, transcription and translation. Which statement about proteins is correct? A beta sheet is indicative of secondary structure. Okay. The complementary strand is wrong. The option is B. COCUUG. mRNA strand of choice is from 5 to 3 is correct. Oh, point mutation causes an amino acid to change to a stop codon. A point mutation causes an amino acid to change to a stop codon. What is the effect on the resulting polypeptide? Is truncated and non-functional. A second point mutation causes that stop codon to change into a codon that is different from the original. It is also the correct answer. So for 25, 9 are correct. Now let's see the second instruction. After the pre-test, we need to do a guided tutoring. Please ensure that for the 30 minutes. So let's start the timer for the 30 minutes and let's get to know here is the answer. It gave the full, it answered all the labels. Nuclear envelope, nucleus, nucleolus, RER, SER, centriole, centrosome, mitochondrion, cytoplasm, plasma membrane, Golgi apparatus, and ribosome. It, it gave the name for every label. Now let's ask the questions. Label G represents centrosome. Cell division. The proteins that assist with cell division, protection, maintenance of cell shape comprise the cell's cytoskeleton. The cytoskeleton is a complex dynamic network of interlinking protein filaments present in the cytoplasm of all cells. And cytoskeleton has been explained. Is composed of three main types of protein filaments: microtubules, microfilaments, and intermediate elements. Let's see the next question. Label F represents, here it has been given label F is centrioles and it is used for cell division. So let's skip this. The organelle represented by label D is mainly found in dash and dash. Nuclear envelope, Golgi apparatus. Label D is RER, no? Is a key component of cells. Okay. Nuclear envelope is associated with two other organelles, nuclear envelope and Golgi apparatus. Okay. The main function of Golgi apparatus is sending and receiving modified proteins. The organelle C is present inside the nucleus of eukaryotic is present in both animal and plants. Yeah, the nucleus is present in both animal and plant cells. Nucleus is a fundamental structure found within the nucleus of almost all eukaryotic cells. Then the organelle labeled is H. That is a mitochondria, and amino acid is a monomer of DNA. What is a monomer of a DNA? A nucleotide is a monomer of DNA. The building blocks of DNA has been given here. DNA is a polymer built from repeating monomers called nucleotides. Deoxyribose phosphate group and nitrogenous base. And it's the here is, we get this also. The sulfate is wrong. The answer is phosphate. Next. DNA is stable than RNA and therefore dash reactive. DNA is more stable than RNA and therefore less reactive. Its instability and reactivity is fundamental to their distinct biological roles and comes down to a tiny but crucial difference in their sugar backbones. Is better suited for its role as a long-term secure storage of genetic information, while the more reactive and transient nature of RNA is ideal for its short-term roles in gene expression and regulation. Okay. This image shows a section of a double-stranded nucleic acid molecule, most likely like a DNA-RNA hybrid. The letters represent nitrogenous bases. And then the best pairing rules. Cytosine always pairs with guanine G. Adenine pairs with uracil when RNA is involved. Delta. This base is on the top strand and paired with U on the bottom. And epsilon ACCA. Bottom strand is UGGU. Okay. And let's ask. According to the strandedness of this molecule, what type of nucleic acid is it? Double-stranded DNA-RNA hybrid. The one strand is DNA. DNA and then according to the types of bases in the molecule, what type of nucleic acid is it? It is RNA. The definitive evidence is the presence of the bases adenine, guanine, cytosine, and uracil. DNA contains adenine, guanine, cytosine, and thymine. Since the molecule in the diagram contains uracil instead of thymine, it must be or at least contain RNA. So, and then what is the paradoxical about this image? The main paradox of this image is that it presents a static, orderly, and spacious depiction of a cell, while a living cell is actually an incredibly dynamic, crowded, and chaotic environment. Let's see. The double strandedness is indicative of DNA. The image shows the chemical structure of ribose in its ring form. The type of sugar is ribose in the ring form. Ribose is a monosaccharide and more specifically pentose ring structure. The image depicts the furanose form of ribose, a five-membered ring containing four carbon atoms and one oxygen atom. This is the common structure of ribose when it's part of biological importance. Ribose is a fundamental building block. It forms the sugar backbone of RNA. So the answer is RNA. What conclusion can be drawn from your findings? The correct conclusion is RNA can sometimes be found as double-stranded. Yes. DNA can sometimes include uracil. The backbone of DNA can sometimes include this type of sugar. The sugar shown was ribose. The amount of hydroxyl groups doesn't matter. We concluded that the extra hydroxyl group on ribose is a critical chemical difference that makes RNA less stable and more reactive than DNA. It matters a great deal. Now let's ask the this one. The ordinary flow of information in a cell is DNA, RNA, protein. The central dogma of molecular biology. The transcription will happen through DNA to RNA and then from RNA to protein. Translation will happen and first transcription and then translation. That is segment of DNA is copied into a messenger RNA. The mRNA molecule then travels out of the nucleus to a ribosome to build a protein. This one also has been answered before, transcription and translation. Which statement about proteins is correct? The correct statement is beta sheet is indicative of secondary structure. The primary structure, it has four levels of structural organization. Primary structure, this is the unique linear sequence of amino acids, and the secondary structure have two types, alpha helix and beta sheet. And the tertiary structure, this is all over. This is overall 3D shape of a single polypeptide chain. The quaternary structure is made up of more than one polypeptide chain. Uh, and then let's ask this one. ADNA. Let's first explain this. This one looks. This image is an mRNA codon chart, a fundamental tool in molecular biology used to translate the genetic code and the specific amino acids they code for during protein synthesis. How it does it work? First letter, find the first base A, and then second letter, find the second base U in the large column on the top, third base in the small row on the right. The key features are redundancy. The code is degenerate, meaning multiple codons can code for the same amino acid. And then start codon. The codon AUG is highlighted because it acts as the universal start codon. The stop codon UAA, UH, and UGA are stop codons. They don't code for any amino acid. Instead, they signal for the termination of protein synthesis. Okay, I understood this. Now let's ask the question for the complementary strand of the given code. A DNA template strand contains this sequence. This is the complementary strand of mRNA, but here is differently given answer. mRNA strand COC yeah, here it is the one 3 to 5. This is the answer. Complementary A adenine pairs with uracil, T pairs with A, C pairs with G, G pairs with C. Okay. Anti-parallel direction 5 to 3 to DNA template strand 3 to 5. Okay. Now let's ask the what and your membrane strand of choice must be reversed before converting to amino acids. That means it must be written in what way? It must be written from 5 to 3 direction. The ribosome binds to the 5 end of the mRNA and moves along the strand toward the 3 end. Okay, Tracy. codon sequential in this direction to build the corresponding polypeptide chain. What protein sequence does this mRNA code for? Notate each protein as its three-letter abbreviation and separate each protein with a hyphen. The answer is GL-glutamine. The translation breakdown, let's see this. The protein sequence is determined by reading the mRNA strand codon by codon and using the genetic code to find the corresponding amino acid. CAG means gel and glutamine. ACU threonine. AUA is isoleucine. GUU is valine. CUC is leucine. I think it is all given in this picture itself. Now let's ask for a point mutation. A point mutation causes an amino acid to change to a stop codon. What is the effect on the resulting polypeptide? The resulting polypeptide will be cut short and it's always non-functional. It's the correct answer. So, what is the effect of a nonsense mutation? This type of point mutation is specifically called a nonsense mutation. Its effect is drastic. Premature stop codon translation halts immediately. This produces a truncated polypeptide. Okay, the explanation is quite good here. Now let's see this. A second point mutation causes that stop codon to change into a codon that is different from the original. What is this mutation called? Let's see this answer. There are some different mutations. Single nonsense suppressor mutation. What is the suppression? Let's ask what are the different types. Mutations. I think there are nonsense, silence, and missense. What is this suppressor? A mutation is a change in the DNA sequence of an organism. They can be categorized based on their scale, their effect on the resulting protein, or where in the body they occur. These mutations often called point mutations. There are silent mutation. The base change results in a new codon, the same amino acid. The change results in different amino acid, then it is missense mutation, which results in a premature stop codon. Then it is nonsense mutation. And then other are insertion and deletion. And the frame shift mutation because of the in insertion and deletion, this is when one or more base pairs are added to or removed from the DNA unless they occur in the multiples of 3 It does cause a frame shift mutation. Frame shift mutation means adding or removing a base shifts from the entire reading frame from the point of the mutation onward. Large scale mutations, these are chromosomal ab- aberrations that affects large segments of chromosomes or even whole chromosomes. Deletion, duplication, inversion and translocation. There are some mutations by inheritance, which are gremlin mutations and somatic mutations. Gremlins occurs in sperm or egg cells. Somatic mutations occurs in body cells. Okay, it explained all the different types of mutations. Now let's see the next. What do we need to do? Guided tutoring. The timer has been. Next, let's set another 30 minutes for this task. For this, we need to ask the model these questions. Okay, let's ask them. What do the nucleus, nucleolus, centrioles, mitochondria, Golgi apparatus, rough and smooth endoplasmic reticulum, cytoplasm, cytosol, cell membrane, cell wall, vacuoles, lysosomes, and customarily look like? Let's see this answer. What are they and how do they look like? Here are the different parts of a cell. Nucleus, the largest organelle, typically seen as a large spherical or oval structure, often near the center of the cell. Nucleolus is a dense and dark spot, while centrioles is a pair of short and barrel-shaped cylinders arranged at right angles to each other. And then mitochondria, it look like oval or bean-shaped organelles, folded inner membrane. These folds are called cristae. Golgi apparatus, a stack of flattened membrane-bound sacs called cisternae. It often looks like a stack of pancakes. Rough endoplasmic reticulum, a network. Oh, let's start this timer. And we got the cytoplasm, the entire content within the cell membrane, but outside of the nucleus. It includes both the cytosol and all the organelles. Cytosol, the jelly-like fluid portion of the cytoplasm that fills the cell and surrounds the organelles. Cell membrane, it is a thin, flexible outer boundary. And then cell wall, a thick, rigid outer layer found outside the cell membrane. Vacuoles, membrane-bound sacs. In animal cells, they are typically small and numerous. In mature plant cells, there is a very large central vacuole. Most of the cell's volume. It explained everything. Now let's see this. What are the functions of these organelles? Cytoskeleton. Are they found in prokaryotes and eukaryotes, animals and plants? What are the functions of all these organelles? Okay, here are the functions of these cell parts and where they are found. So, first, nucleus. The function is it is a control center. It contains the cell's genetic material DNA and controls all cell activities. It found in eukaryotes only, but found in both animal and plant cells. Nucleolus is located inside the nucleus. Its sole purpose is to manufacture ribosomal RNA and assemble ribosomes. It, it may found in eukaryotes only, but found in both animal and plant cells. And then centrioles, the function is they organize microtubules to form the spindle fibers that separate chromosomes during cell division. It may found in eukaryotes only and found also animal cells only. Animal cells but are absent in most higher plants. Okay, mitochondria. It functions as the powerhouse of the cell. They perform cellular respiration to convert glucose and oxygen into ATP, the main energy currency of the cell. Okay, it found in eukaryotes only, but found in both animal and plant cells. And then Golgi apparatus, the post office of the cell. It modifies, sorts, and packages proteins and lipids into vesicles for transport to other destinations. Found in eukaryotes only, found in both animal and plant cells. And then rough endoplasmic reticulum function, its attached ribosome synthesizes proteins that are destined for secretion or insertion into membranes. It also modifies these proteins. Found in eukaryotes only, found in both animal and plant cells. Cytoplasm. Suspend. It serves to suspend the organelles found in both prokaryotes and eukaryotes and in animals and plants. Cytosol. This functions like a jelly-like fluid portion of the cytoplasm. It provides a medium for biochemical reactions. It found in both prokaryotes and eukaryotes and in both animal and plant cells. Cell membrane, also called plasma membrane, is semi-permeable barrier that regulates which substances enter and leave the cell. Okay. Cell wall functions has been given provide rigid structural support protection gives the cell a fixed shape. Most prokaryotes and some eukaryotes. It may found in most prokaryotes and some eukaryotes. It is found in plants but not animal cells. Vacuoles primarily for shortage of water, nutrients, and waste in plants. The large central vacuole also provides turgor pressure for structural support. Found in eukaryotes only, found in both animal and plant cells, but they differ. Plants have one large central vacuole, lysosomes, the recycling center. They contain digestive enzymes to break down waste materials, foreign invaders, and old worn out organelles. It may be found in eukaryotes only. And lastly, cytoskeleton, a network of protein filaments that provides structural support, maintains cell shape, enables cell movement, found in both prokaryotes and eukaryotes and both animal and plant cells. Now let's ask the next question. What is the difference between DNA and RNA in terms of nucleotide composition, stability, base composition, characteristics, process that make it, and process they undergo? Let's see the answer. Nucleotide composition: DNA composed of a phosphate group, a deoxyribose sugar, and a nitrogenous base. And RNA is also composed of phosphate group and a ribose sugar and a nitrogenous base. Only difference is the deoxy and this both. And then coming to the stability, DNA is highly stable. The absence of the reactive 2 hydroxyl group and its double helix structure make it resistant to degradation, while RNA is less stable and more reactive. Base composition uses 4 bases in DNA: adenine, guanine, cytosine, and thymine. In RNA, in place of thymine, it uses uracil. The standard base pairing for DNA is AT and GC. For RNA, it is AU and GC. The characteristics means the structure and function. DNA structure is double stranded. It serves as a permanent blueprint for life. Okay. RNA is a single stranded. Acts as a versatile worker molecule. Process that make it: DNA replication, RNA made through transcription. Thus, RNA undergoes replication during cell division and transcription to have its information copied into RNA. The messenger RNA molecules undergoes translation where its code is read by a ribosome. Okay, let's see the next question. Uh, the consider the Consider this template strand of DNA. How does the information transform at each step as it becomes a protein? Include the anticodons. The information in the DNA strand is transcribed into mRNA Consider this template strand of DNA. How does the information transform at each step as it becomes a protein? Include the anticodons. The information in the DNA strand is transcribed into mRNA and then translated codon by codon using tRNA anticodons to assemble a specific amino acid sequence, which for the strand has been. uh, Let's see this one. What are the different types of mutations? Here it gives the full information about the DNA strand. Now let's see the different types of mutations. A mutation is a change in the DNA sequence of an organism. They can be categorized based on their scale, their effect on the resulting protein, or where in the body they occur. These mutations are often called point mutations. There are three types here: silent mutation, missense, and nonsense mutation. The other ones are insertions and deletions, which means added or removed from the one or more base pairs are added or removed from the DNA, and then frame shift mutation because DNA is read in three letter codons. Large scale mutations, these are chromosomal aberrations that affect large segments of chromosomes or even whole chromosomes. Deletion, duplication, inversion, translocation. Okay, now let's see generate. Okay, let's add this file. Now let's see. Here are the questions. What is the main function of the organelle labeled K? Practice questions. K. Let's see the image. K is Golgi apparatus, no? Then answer will be, I think, A. 2 is a mutation changes a DNA codon from 3AGT5 to 3ATT5. What is the name of this? Think. I think it's B. Let's put B. Third one is in the diagram of the nucleic acid, what nitrogenous bases do represent? A DNA monomer is composed of a nitrogenous base. What is this? Let's answer sixth one. That made up of the structure labeled G, or known as G, is B. Let's answer eighth one. The structure labeled F with centrioles. Oh, here are the answers. Why they are wrong? Okay. What is the main function of the organelle labeled K is modifying, sorting, packaging proteins and lipids. It is a nonsense mutation here. And then 3A. 3A only I answered, no? Adenine and uracil. And then for 6, it is cytoskeleton. Yeah, that is what I wrote. And then 7th, I didn't answer. For this, the beta sheet is a common type. Yeah. And then centrioles and cell division. Why it is a fundamental paradox. Double standard. Okay. We have done this also. Now let's see the next step. By the next step, we need to do a post test. Let's see the post test. Here, use this diagram to answer the following 6 questions. A planter has been given and some labels has been given, and the first question is label 6 represents an organelle. Label 6 organelle. What is it called? That produces sugar molecules. What produce sugar molecules? Isn't it mitochondria? Label 4 represents dash, which is necessary for. Label 4 is, I think this is the large, no? So I think it's central vacuum. Let's put this maintaining turbidity. And then label 10 is the internal layer of a substance that surrounds the cell. This is called the cell membrane. Right? And the main function of the smooth endoplasmic reticulum represented by let's put this lipid synthesis. Label 5 is present in label 5. Let's put some eukaryotes. The organelle labeled will be in higher numbers in muscle cells compared to fat cells. Let's put 8. The backbone of polymer ribose sugar. Ribose sugar will be in RNA. Sulfate is wrong. Phosphate is the correct answer. Why is RNA not the main carrier? Is single stranded also, no? Use this diagram to answer the following 7 questions, as it looks like a single strand, so let's put RNA. According to the types of bases in the molecule, what type of nucleic acid? I put DNA. What is paradoxical about this image? Don't know. According to the question 11, Is this question 11? Ribose sugar. Ribose, water, and maybe A and T. What conclusion can be drawn from your findings? A genome that is comprised of single-stranded DNA is as sufficient as double-stranded. Next, reverse transcription means uh, RNA to DNA. The inverse of the process described in the previous question is RNA synthesis. Which statement about proteins is correct? A straight chain of amino acids is indicative of primary structure. Use this diagram to answer the following three questions. Uh, GTA. G will be modified with C, A with U. I think this is the answer. Now, what is the most likely reason that the sequence in the question has been written in reverse order from the answer? What protein sequence does this mRNA code for? Separate each protein. CAU. CAU is his, and then AAC. AAC is ASN, and then UUU produce PHE, and then CGA. CGA is ARG, and then CAC. CAC is his. I think that is one. Now let's see this. A point mutation has occurred, but the polypeptide is the same. I think it is nonsense. And then a point mutation occurs in a separate polypeptide. The second one interacts with the first one in an to become truncated. I think makes sense. Let's submit this. Let's check the score. 14. The label 6 is chloroplast. Okay. That is what I am. Membrane is correct only, no? Okay. And this one is 9 lipid synthesis also correct. The organelle label will be in higher numbers in muscle cells is 1. 1. Okay. And then a DNA monomer is called a nucleotide. Oh, it's deoxy, no? Oh god. It's deoxyribonucleic. These both are wrong and this is the correct. Oh, sorry. Why is RNA not the main carrier? Is less stable than DNA. It's also correct, no? Okay. And it is RNA and then nucleic acid DNA. It's correct. The paradoxal about this image is the single stranded nature indicates RNA, the ribose sugar and deoxyribose. What conclusion can be, and for this one, A and T respectively, and the conclusion, this one is also correct. That is what I wrote, no, RNA, DNA. I think this one is also correct. 2 has been given wrong. The inverse process in the previous question is transcription. Which statement about proteins is correct? A straight chain. It's also the correct. And a DNA template. Yeah, we got this. Those 3 are correct. Oh, point mutation. Sorry, it's a silent. No. And then here is the nonsense. That's it. Let's stop the